Hello and welcome to the 12th episode of the Innova Biosciences webinar series. Today's flow cytometry webinar will discuss sample preparation and the process of experimental design, as well as hints and tips that will help you set up a successful flow experiment. Our speaker today is Dr. Andy Lane. Andy will discuss cell source and preparation, sample treatments, controls and maximizing signal, and choosing and using antibodies. There will be a question and answer session at the end of the presentation. Please submit your questions by typing in the questions box in your control panel, which is circled in red here. Feel free to send questions throughout the webinar and we will answer as many as we can in the Q&A session. Any remaining questions will be answered by email in the next few days. So that's the basics laid out. Now please welcome Andy.
Thank you very much, Claudina, and welcome everybody to this webinar. As Claudina outlined, I'm not really going to spend much time talking about the technical aspects of flow cytometers themselves today, or anything such as filter arrangements or the lasers. I'm going to be thinking more about the samples themselves and what you need to understand about preparing your samples for running on the flow cytometer. Quite a lot of what I'm going to talk about is the cell samples themselves, and obviously, as this slide picks out, one of the key requirements for flow cytometry is that the sample is a suspension of particles. We're going to be talking about cells today, but it could also be bacteria, it could be other particles, etc. As I mentioned, understanding the nature of the sample is key to designing your flow cytometry experiment. You need to know exactly what sample you're working with and how it is going to behave biologically or with the antibodies, etc., that you're staining with. One of the key aspects is, do the cells that you're working with need to be selected or prepared in some way? I'm going to run through a number of different cell types, and perhaps one of the most obvious ones to start with is whole blood. This, particularly for people working with human systems, is one of the most common samples to stain. So whole blood, as I'm sure you're all aware, is a mixture of leukocytes, and there are 5 main types here: lymphocytes, monocytes, neutrophils, eosinophils, and basophils. From a flow cytometry point of view, quite often neutrophils, eosinophils and basophils are lumped together and called granulocytes. That terminology covers those 3 together, but the most important ones in terms of numbers are the neutrophils. Remember, there are also platelets within the whole blood sample, and of course erythrocytes, a huge number of red blood cells, which, although in many cases are removed, are there as a key component of your sample. You need to understand the antigen expression on each of these cell types. When you're staining with a particular antibody, you need to have an idea of which cells you expect to be stained and where you're going to find them on a scatter profile. Remember that if your antigen is expressed by the red blood cells, by the erythrocytes, you'll need probably a much larger amount of antibody than would be normal, since you already have a large number of red cells that will be expressing that marker. They will effectively soak up that antibody. CD59 would be a good example, which is expressed by red cells, but also by all of the leukocyte subpopulations. When you're working with whole blood, there are still a number of things you need to consider, and some of them are not quite so obvious. One of them is the anticoagulant that is used. So obviously you need to have an anticoagulant in your system to make sure that the blood doesn't clot. The 2 most common ones are EDTA and heparin. Sodium citrate is another common anticoagulant. People are often not aware that some of the anticoagulants can have an effect on the markers that you're looking at. A primary example, for instance, can be CD41, which is a platelet marker, and that some antibodies recognizing CD41 are sensitive to the presence of EDTA. So you would get a false negative result in the presence of EDTA. You need to be aware of factors such as this and choose the correct anticoagulant for your sample and for the staining that you're undertaking. You also have a choice of the lysis solution, the solution that you're going to use to remove the red blood cells after staining. Perhaps the original one was ammonium chloride, which is still quite commonly used, but there are now many commercial solutions that actually fix the cells at the same time as lysing the red blood cells. Once again, be aware that certain formulations just occasionally cause problems with individual antibodies, and you need to be aware of that and look for manufacturers' recommendations. If you're using an antibody from a particular manufacturer and a lysing solution from them, you should be quite confident. But if you're mixing things up and you get strange results, it's always worth just double checking that the antibody is compatible with that lysis solution. Most lysis solutions will work with all species, so therefore the same solution that will lyse human red blood cells will lyse mouse and rat red blood cells. Do be aware of nucleated red cells in some pathological samples, and remember that avian samples, so bird red cells, are nucleated and they will not be lysed by the normal lysing solutions. So don't be surprised if you have problems if you're using samples of that sort. Another thing to consider with whole blood is, are there any interfering substances in the plasma? The primary example here is the example of immunoglobulins. There are a very, very high level of immunoglobulins in the plasma. And if you are looking to stain surface immunoglobulin on B lymphocytes, the soluble immunoglobulin in the plasma will actually block the staining of the antibodies simply because there is a huge excess of it. This is simply overcome by washing the red blood, the red cells, blood sample, so that you remove the immunoglobulin before staining. The simplest way of doing this is washing it 3 times in a warm isotonic buffer. Moving on from whole blood, the next most obvious sample to talk about is something called PBMCs, or peripheral blood mononuclear cells. This is a subset of the leukocytes in whole blood prepared using a Ficol centrifugation gradient. You can see in the picture on the right-hand side what happens when you lay a sample of blood over Ficol and then centrifuge that, usually for about 20 minutes or so. The red cells pellet at the bottom, the plasma is seen as the yellow color at the top, and there is a band of mononuclear cells, the lymphocytes and monocytes, exactly where the arrow is pointing. And you can remove these very gently with a pipette, and you have a purified population of cells. This process is quite often used when you want to isolate these cells for further work, perhaps for some cell culture analysis, etc. Obviously, you can use these cells for flow cytometry, but you should also be aware that because you've been isolating a particular subset of cells, certain cell subsets might not be there in exactly the same numbers as they were in the whole blood. You may get some losses. It's generally not a major problem, but something that you should be aware of as a possibility. Other than the PBMCs, you can also work, and many people do, with cell lines. So immortalized cell lines, of which there are really now dozens and dozens of particular types. You can have B cell lines, T cell lines, macrophage cell lines, etc. One key thing about these is that you get a homogeneous population. You don't really need to separate out cells with scatter gates, etc. But do be aware that cells within a population will be at different stages of the cell cycle, and depending on how well the culture is going, the viability of the cells may vary. You may indeed see 2 separate populations if there are a high number of dead cells in your sample. One thing to be aware of is that you may have different results if you're using cell lines from different sources. So for instance, the well-known T lymphocyte cell line JERCAT has a number of different subclones, some of which express CD3 quite strongly, some of which express CD3 very weakly indeed. So although you may expect a certain result, be aware of the provenance of the cell line that you're using. Moving on from blood samples and samples that are relatively easy to prepare in a single cell suspension, it is also possible and quite common to undertake flow cytometry on cell suspensions that are prepared from solid organs. Probably the typical example come from the immune system, so lymph node and spleen, etc. But effectively you can prepare cell suspensions from any organ. There are two main ways of doing this. One may be physical disaggregation, or you can assist that with some enzymes, particularly collagenase. Physical disaggregation usually involves literally mashing the organ up, often with the assistance of a syringe. Collagenase helps to break down the connective tissue and releasing cells from within the organ. Be aware that if you are using an enzyme such as collagenase, there is the risk that the enzyme may damage cell surface molecules, and therefore it may be good to culture those cells for a few hours after disaggregation so that those markers may be re-expressed. One thing to be aware of is that not all cells are released from tissues in the same way. So if you take a spleen sample and disaggregate that manually, you will find that lymphocytes are released very easily, but that macrophages tend to be, shall we say, stuck within the tissues. And therefore you may get a difference in result between your flow cytometry staining and immunohistology staining when you're looking at tissue sections. A key example would be the well-known macrophage marker F480, which stains a lot of cells when looking with immunohistology and relatively few cells when looking at a cell suspension from a spleen. This is often because many of the macrophages are actually caught up in the connective tissue of the spleen and not released effectively. Once you've got your cell preparation, you need to consider the location of the antigen on the cells. Put simply, is it at the cell surface, in the cytoplasm, or perhaps in the nucleus? If it's from within the cell, one thing to consider is whether or not it's a secreted molecule. The key example here would be cytokines, which are generally measured as in the cytoplasm of cells, but of course they're always being released by the cells on an active basis. If you want to increase the concentration of the cytokine within the cell, you can use a transport inhibitor such as monensin or Brufeldin A to increase the levels of the protein. This often involves a pre-incubation of perhaps 4 hours before you undertake your flow cytometry staining. Be aware that some cell surface antigens have epitopes that are intracellular. So a good example here would be CD3, the T cell marker. Many antibodies for CD3 recognize cell surface epitopes, but some of them, particularly those that recognize a whole range of different species, recognize an epitope of CD3 that is within the cell membrane, that is intracellular, and you need to use permeabilization techniques to stain that marker. So you need to know which epitope your antibody is going to bind to. If you're looking at intracellular staining, you need to be able to permeabilize the cells to give access to antibodies to get to inside the cell. Generally, you need 2 stages, a fixation step to stabilize the cell membrane and a detergent step to actually permeabilize the cells. The original reference method was fixation of the cells with paraformaldehyde and permeabilization with the detergent saponin. If you're using that method, be aware that in many cases the detergent needs to be included throughout the staining procedure. Therefore, if you're undertaking, for instance, an indirect stain, you need to keep saponin in your wash buffers to ensure that the effectively the holes in the cell membrane are maintained throughout. Staining of nuclear antigens might require additional steps, such as further methanol treatment. In many cases these days, people are using a whole range of different commercial reagents for permeabilization. And my recommendation there would be to follow the instructions that come with the antibodies that you're using wherever they're available. It's quite possible to combine staining of cell surface markers with intracellular staining. Again, perhaps the most common example would be staining cytokines within lymphocyte subsets. So for instance, you might want to look at CD4 and CD8 cell subsets using cell surface markers and cytokines such as interferon gamma, interleukin 2, etc. Within the cells. Consider the order of adding your antibodies if it's going to be relevant. You may need to stain the cell surface antigens prior to permeabilization. And also be aware, particularly if you're using some formulations for nuclear staining, that some fluorochromes may be damaged. So for instance, a methanol protocol for undertaking nuclear staining may well damage phycoerythrin conjugated antibodies that have been used to stain cell surface markers. So once again, plan your experiments and understand exactly what you're looking for and the impact of your reagents on the antibody staining that you're undertaking. Controls are obviously a key part of any experiment, not just a flow cytometry experiment. And again, this comes back to the real basics of experimental design. We need to think both about negative controls and also positive controls. The aim really is to differentiate between true positive signals and non-specific binding of antibodies and or autofluorescence of the cells. So a key control should be just to use the cells that you are working with no additional reagents added. This will give you the baseline fluorescence of the cell population being studied. It will pick up any autofluorescence, perhaps from dead cells or other cells that are going to show staining in particular fluorescence channels that you're working with. Another useful control is the isotype control. This is a monoclonal antibody from the same species, the same subclass, with the same fluorescent dye, and hopefully at the same concentration as the test antibody you're working with. One thing to be aware of is that the isotype control is really aimed at identifying non-specific binding. It won't necessarily give you the same result as the cells-only control mentioned earlier. And a good example of this may be that if you're working with a cell population containing monocytes or macrophages that have high levels of Fc receptors, the isotype control will identify those cells and will show Fc-related binding. This is the control doing exactly what it's meant to do. And in some cases people say, I'm getting a very high result with my isotype control, and that really is identifying a specific nature of that cell sample that you need to be aware of. So if you look at this set of figures, you will see a couple of these controls, in this case not showing a great deal of difference. In step 1, we're gating on the lymphocyte population. Step 2 is literally using just those unstained cells. So here there are no antibodies, etc. present. Step 3 are the same cells with an isotype control. Here you're not seeing any isotype control binding, but if these were a macrophage population, for instance, you may see Fc-mediated binding and a higher apparent staining than you see with the cells-only control. And then in step 4, you've added your specific antibody, and you can see a positive result. There are more controls to consider. Over the last few years, the importance of the so-called FMO controls, or fluorescence -1 controls, have become much more widely known. These are particularly important in multicolor experiments. They help you with setting compensation, and they also make sure that you're not seeing any other non-specific binding from other antibodies within your staining panel. Remember, staining panels now are regularly perhaps 6, 8 or 10 antibodies, and the opportunity for non-specific binding in your samples is much increased. The FMO control is literally every antibody in your panel apart from the color that you're currently controlling. So you may remove the FITC antibody from a panel of 5 or 6 other antibodies to set the control for the FITC channel. Positive controls are often the ones that are most easily overlooked. People think a lot about the negative controls, but remember that it's good to be able to confirm the activity of the reagents that you have. If you're working with simple samples, perhaps with lymphocytes, you often have a lot of internal controls of T-cell markers and B-cell markers that are staining as you might expect. But if you're looking for activation markers, you may need to be convinced that your cells have been actually activated in the way that you would expect. So you might want to use a broad activation control to show the activation status of the cells. For instance, CD25 or CD69 can be used as activation markers to show that cells have been activated as you expect them to have been in the earlier parts of your experimental protocols. So we've thought a little bit about controls. Obviously, one of the things that you want to do with flow cytometry is to maximize your signal. One of the ways of maximizing your signal is to reduce your background, and there are a number of ways of working to do this. One of the key things to do is to make sure that you always have a dead cell dye to exclude dead cells from any analysis that you are working on. Dead cells tend to have high levels of autofluorescence, and they may possibly also have aberrant antigen expression. So it's very good to be able to exclude dead cells from your system. There are a number of dead cell markers available now, and they can generally be found for use in whichever channel of the flow cytometer you need to find a dye to work with. So you're not just restricted, perhaps as you were a few years ago, to something like propydium iodide. There are dead cell markers now available for all channels. You may want to consider the use of blocking antibodies. These work basically by pre-incubating your cells in the serum of the same species as the antibody that you're using. Probably particularly important in indirect systems, but you can use them with direct systems as well. A particular example of blocking antibodies is the well-known use of so-called FC block in mouse systems. FC block can really be considered as a more defined version of using serum, as I mentioned above. The prototype antibody for FC block is a clone called 2.4G2. It recognizes mouse CD16 and CD32, which are FC receptors. And this antibody, when used in an unlabeled format, blocks FC-mediated binding to the FC receptors. And then when you add your directly labeled primary antibodies, they will only bind to their specific antigen and not to the FC receptor. Following on from 2.4G2, there are now a number of other monoclonal antibodies of the same specificity that are available from a range of other suppliers. So you have a lot of choice in determining how to undertake your FC blocking. Perhaps the major consideration in terms of reducing background is to actually titrate the antibodies that you're using. This is one of the things that may sound very obvious, but in my experience is one of the things that people are actually not doing as effectively as they may do. Manufacturers will generally give you an indication of the dilution or the concentration of the antibody that you should be using. However, remember that they will have determined that in a specific set of conditions, and it is always best to repeat those experiments with your own conditions. You may well be able to dilute your antibodies much further than a manufacturer recommends, which may well reduce your background, and it may well, of course, save you a considerable amount of money at the same time. Also consider the fact that not all antibodies perform optimally at the same concentration. So it is not effective simply to use all of your antibodies, for example, at 10 micrograms per mil. If you have a very high affinity antibody, you may be able to use it at a very, very high dilution indeed, and in comparison to a lower affinity antibody, which may need to be used at a significantly higher concentration. Therefore, titrate all of your antibodies. If you're using antibodies at a higher concentration, remember that at some point will not increase your positive signal. The positive signal will remain where it is, and all you will get is an increased background stain. So, as I said, titrate your antibodies to get the best result, the best positive signal, but also to minimize the background that you have. As well as titrating your antibodies, think about the staining conditions of your cells, so the volumes that you're using and the temperature, etc. It's always best to keep the volume that you're undertaking the staining reaction in as low as possible. If your cell density is very low, it's much better to concentrate the cells by centrifuging them than to use a high volume for the staining step. So most staining is probably carried out in 50 to 100 microliters, which is absolutely fine. I certainly wouldn't recommend trying to undertake your staining in half a milliliter or 1 milliliter, even if you think you're using the same number of cells. Remember there, effectively your antibody will be very dilute, and the reaction with the cells will be significantly reduced. It will certainly be much slower. Also consider the incubation time and temperature. You will find many different opinions over staining on ice or at room temperature, and also on how long you should stain with your particular antibody. I think in many cases people recognize that with most antibodies the difference between a 15-minute and a 30-minute incubation is quite small, and there is probably little difference between staining on ice and at room temperature. But do be aware that some antibodies are much more sensitive to those conditions than others. I can certainly remember an antibody to the mouse T-cell marker CD3, which I have used in the past, which is much more effectively stained at room temperature than on ice. If you get to know these characteristics of the particular antibodies, you can significantly improve your staining results. The key thing to remember is that your antibody binding is a dynamic event. Remember that antibodies will bind onto their antigen, but then they will also have an off step from the antigen, and that is a cyclical event. Effectively, there are on and off steps ongoing all of the time. You want to set your reaction conditions to maximize the on binding of the antibody and minimize the off step. That's one of the key considerations with the volume of staining. If your volume is too high, the on step will be much slower and it will be much reduced. What about selecting antibodies? Obviously, in terms of specificity, you now have a huge choice, both available commercially and for many of you in your own laboratories. When you're thinking of flow cytometry, you're really thinking of what color you want to use each particular antibody with. Perhaps the general principle is to use the brightest fluorochrome for the most weakly expressed antigen, the so-called stain index. There are various different sources of tables of specific fluorochromes which show which fluorochromes are brighter than others. I'll mention some sources of information in just a moment. Effectively, markers such as phycoerythrin are known to be very bright. Markers such as fluorescein are less bright when staining the same antigen, and I'll show you an example of this right at the end of the webinar today. As well as brightness, you need to understand the capabilities of the flow cytometer you are using. May sound fairly obvious, but which lasers does it have? Which lasers should you be using for the particular fluorescent markers that you have available? And also what filter sets are fitted or are available for retrofitting to your instrument. Again, take advice from your flow cytometer manufacturer on these particular aspects. When you're undertaking multicolor experiments, choose dyes that minimize the spectral overlap. There are various sites, and I'll mention some in just a moment, that have excellent sources of information about emission spectra of different fluorochromes. As you see from the diagram, this is a simple diagram of fluorescein overlapping with phycoerythrin, but fluorescein can overlap into the phycoerythrin channel and vice versa. This is where the compensation requirements come from. These two markers don't overlap a great deal, but if you're undertaking multicolor analysis with some of the dyes that overlap much more closely, you want to consider carefully how you design your experiment. I've mentioned some online resources, and I was just going to highlight 3 or 4 sites that I find particularly useful in this respect, that many of you may be aware of, and all of these are certainly worth looking at when you're thinking of designing your flow cytometry experiments. Chromosite.com, Flourish.com, and Cytogeny on the woodsidelogic.com address all have tools that will help you to design your experiments. It will give you details of brightness of different fluorochromes and the overlap and the compensation that you expect between those different fluorochromes. Related to overlap of fluorescent spectra, the chroma.com website has a very useful tool that allows you to pick the particular dyes that you are using and to see how they overlap together in terms of emission spectra, etc. This isn't an exhaustive list of resources, but it's certainly some of the most useful that I've come across. When you're designing your experiment, you obviously may well come across the situation in which the antibody that you need isn't available in the color that you would like it to be in for your experiment. With the increasing number of commercially available antibodies, you will find some still that do not have the right color available when you're moving up to 5, 6, or 7 colors or even more in flow cytometry. So you may be able to find the antibody but not in the color that you need. You may of course have your own antibody that you need to be conjugated to a particular color for a multicolor experiment. So your choices at this stage are probably to have a custom-made conjugate prepared, either from a commercial antibody, or to prepare your own conjugate using traditional techniques. Both of these can be quite time-consuming, expensive, and utilize a lot of antibody. The Lightning Link conjugation kits from Innova Biosciences offer a very quick and easy-to-use solution for preparing such conjugates, and actually allow you to use as little as 10 micrograms of antibody when preparing conjugates for your experiments. So if you have particularly expensive or valuable antibodies, you can undertake conjugation on a very, very small scale indeed. So I'd just like to talk for a few minutes about what Lightning Link technology is and how you can use it, and show you some of the results obtained. Effectively, Lightning Link offers the fastest, easiest to use and most efficient conjugation technology available. It literally takes 30 seconds to label an antibody with a fluorescent dye or an enzyme. There are more than 50 labels available, and many of these are relevant to flow cytometry, being fluorescent proteins of different wavelengths. When you're undertaking a labeling reaction with Lightning Link, literally the only equipment you need is a pipette. You take your antibody, you add a small amount of modifier reagent to that, and immediately then use that material to reconstitute a freeze-dried pellet of fluorescent dye. You can then leave that to incubate for either 20 minutes or 3 hours, depending on the format of the kit that you're using. And at the end of the reaction, you simply add a quencher reagent, and your reagent is then ready for use, basically to titrate in your experiment. There is no need to undertake any further purification. You don't need any centrifugation or spin steps or chromatography columns to separate out unlabeled dye. You can use the antibodies immediately as they are in your experiment. Because of the lack of spin columns, etc., you have 100% antibody recovery, so you can use your very small samples and know you still have plenty of antibody for the test that you need to do. You can actually scale your reaction from the very small scale, 10 micrograms, up to much larger scales, and we offer kits for up to 1 to 5 milligrams, and we can go much higher than that if people have those sorts of requirements. Conjugating 1 milligram of antibody will provide you with many thousands of tests for most flow cytometry experiments. The simplicity of the reaction overcomes batch-to-batch variability, and as well as labeling antibodies, which we're mainly focusing on today, you can label other proteins or biomolecules. You have a very stable reaction because of the covalent bond, and as I mentioned, you have 2 different formats of the kits. Our rapid kits, which have a sort of 15 to 20 minute incubation, and our standard kits have a 3-hour incubation. The lightning link range in terms of dyes for flow cytometry is quite extensive, and there's a selection of them shown on the table here. They've been grouped by the most commonly used excitation lasers. So there are a couple there working off of the 405 nanometer laser line, a lot of dyes working off of the most common 488 nanometer line, shown there in green. In yellow, dyes that will work very effectively off of some of the newer lasers, perhaps the 561 laser line or even the 532 laser line. And then those off of the 633 or 635 laser line. So you'll see you have a wide choice of dyes here, which are perfect for fitting into your multicolor flow cytometry experiments. There are just a few things to think about for the antibody that you're using. As I said, the reaction itself is very simple. The antibody, though, should be at 1 milligram per mil. If it's much lower than that, the efficiency of the reaction may well be reduced, but we have very simple kits available as well to concentrate antibodies. It's important that your antibody is as pure as possible, so ensure that it's been purified, and also make sure that stabilizing proteins such as BSA haven't been added back into your antibody preparation, as they will interfere with the reaction. Most common buffer formulations are suitable. PBS is probably the most common, and that is absolutely fine to use. But do make sure that amines such as glycine are absolutely absent from the reaction, and also that thiols such as DTT and mercaptoethanol are not present. A small amount of tris has no effect on the reaction. As I mentioned before, it's possible to use lightning link with antibodies, but also with some other proteins. I do have just a couple of examples of how lightning link works and how it compares to commercially available antibodies. So for instance, in the example shown here, we've undertaken a comparison of lightning link staining of the CD4 cell surface marker using lightning link fluorescein with a commercially purchased antibody from the same clone staining CD4 cells. And you'll see that exactly the same staining intensity is seen using lightning link as with the traditional antibody staining. In this particular experiment, we're looking at another marker at CD54. Now, CD54 is much more weakly expressed at the cell surface, and this is a good example of where you can see the difference between the very bright phycoerythrin marker and the less bright fluorescein or FITC marker. At the top, you'll see phycoerythrin staining. Top left, a directly conjugated antibody purchased from a leading antibody manufacturer. In the middle at the top, the lightning link conjugate, and overlaid top right to show these are exactly the same result. And then using fluorescein, you will see that actually taking the same approach of a commercial antibody with a lightning link conjugate, you'll see exactly the same results, but very, very weak staining, simply reflecting the fact that fluorescein is a much less bright dye. So those results demonstrate how the lightning link reactions give you exactly the same performance as you will obtain with commercially obtained antibodies. So I'm going to stop there. I hope that some of this has been useful. I'm going to pass back to Claudina, and I think we see some questions coming in, and we'll try to answer some of those in just a moment.
Okay, thank you very much, Andy. Well, let's move on to our Q&A session. So there's still time to submit questions. Simply type them in the questions box as shown in the picture on the slide. And thank you to everyone who already submitted questions during the presentation. So Andy will now answer as many as possible in the time allocated, and any remaining questions will be answered by email. But I would just like to very briefly tell you about where you can find us in the coming months if you would like to chat to an Innova representative in person. So in early April, we will be exhibiting at the American Association for Cancer Research annual meeting, which takes place in San Diego. And in early May, we will be at the Immunology annual meeting in Pittsburgh. So please feel free to stop by our booths, or alternatively contact us if you would like to set up a meeting. Okay, so I believe Andy is now ready to answer some questions. So, right, our first question today is, should I expect to see any differences when performing flow cytometry experiments with samples from different species?
Okay, thanks, Claudina. So I think the answer to that really is, in some cases, yes. For example, the scatter profile of whole blood when you're using rodent samples will look much different to the scatter profile of human blood prepared using the same lysing solution. Generally, rodent samples have a much less clear position of the granulocytes, for instance, on the forward and side scatter profile. Possibly another important point is to be aware that antigen expression may sometimes differ between species. Probably the best example I can think of is CD4, which in humans is known to be expressed on a subpopulation of lymphocytes and also on monocytes, but is definitely not expressed on neutrophils. However, in the dog, you will see CD4 expressed on neutrophils, which the first time you see that may be a little bit of a surprise, but it's a very real result.
Okay, thanks for that, Andy. So our second question is, do I need to use an FC blocking reagent when using human samples in the same way as with mouse samples?
Okay, so generally I would say that FC-related binding tends to be less problematic in human samples than in mouse samples. One reason for this is that a high proportion of the samples of human material used in flow cytometry are undertaken using whole blood. And in such cases, the immunoglobulin present in the plasma, as I mentioned during the talk, will already be occupying FC receptors present on the cells, such as monocytes. In human samples, there is a high affinity FC receptor known as CD64, and that preferentially binds mouse IgG2A antibodies. One option to block this is to include an unlabeled, irrelevant mouse IgG2A before staining with a direct conjugate. However, there are now also some commercially available reagents that will reduce FC receptor binding in human samples.
Okay, so our next question is, the antibody that I want to label contains BSA and I only have a small amount of it available. You mentioned that the lightning link kits will not work as well with BSA present, so is there anything I can do?
Okay, yeah, there's quite a simple answer to that. Along with the labeling kits that I mentioned, we've got a number of kits that enable you to either purify or concentrate very small volumes of antibody into a format suitable for conjugation. In terms of BSA, we specifically have a kit designed to remove BSA, which is often present as a stabilizer in antibody preparations. You can see full details of those on our website, or we can send details if people want to ask individual questions about those reagents or kits.
Okay. And our next question is, how long can I keep an antibody following labeling? I will assume that means with the Lightning Link kit.
Okay. Generally, conjugates made with Lightning Link are going to be stable for a long period. I think I mentioned it's a covalent bond. It's a very stable bond. It's probably important to reiterate that you should always treat your antibody conjugates with respect and care. We recommend that you should store them at 4 degrees and keep them in as concentrated a form as possible. In most cases, you would probably expect stability of at least 12 to 18 months, possibly longer. Be aware that some dyes are perhaps inherently less stable than others. The ones that really come to mind here are the tandem dyes, which are somewhat less stable inherently, and they also may be particularly sensitive to light. But generally, a Lightning Link conjugate will be stable for quite a long period after it's been made.
Okay, and our next question is, I have a protein that I am using to study receptor binding, and it would be really useful to link this to a fluorescent dye. Is this possible?
Most likely, yes. I think I mentioned that you could use Lightning Link kits to label other biomolecules. Proteins binding to receptors would fit into that category. The key thing is that the protein needs to have a primary amine group available for the conjugation reaction to take place. That's going to be the case with most proteins, and thereafter the main criteria is to adjust the reaction conditions according to the size of the protein that you're working with. So Lightning Link kits are designed to use with antibodies. If you have a protein that is half the size of an antibody, then you should use half the amount of material that you would typically use with an antibody for that product. For more specific details on conjugating your particular protein, I'd suggest you contact us directly and we can give you some advice about exactly how to adjust the reaction conditions.
Okay, thanks very much, Andy. I'm afraid that's all the time we have today for questions. However, as I previously mentioned, we will respond to any remaining questions by email. So please bear in mind that this webinar is recorded, so we will upload it to our YouTube channel in the next few days. You can find our channel at youtube.com/Innova Biosciences. And also please keep an eye out on our social media channels where we will soon be advertising our upcoming webinars. And if you would like to receive a PDF copy of today's presentation or a link to the recording, please contact me. You can find my contact details on any of the webinar emails that you've received. So thank you very much for joining us today. We hope that you enjoyed the webinar, and we hope that you will be able to join us again for the next webinar in March.